Hey everyone, welcome back to another episode on genetics. Today in this chapter, in this section, we're going to go over the three different types of proposed mechanisms for DNA replication. But to begin with, these are the three ones, semi-conservative, conservative, and dispersive. This one turned out to be the correct one. And then the other three were not correct. But to begin with, semi-conservative is essentially the model that predicted that if these two parental strands were replicated, you would end up with a duplex that had one parental strand and one newly synthesized strand. So we can say that the red chromosomes are the parental strands, and then we can say the the blue chromosomes are the newly synthesized strands. So after replication, what will happen is that you would end up with a chromosome duplex that had the original parental strand, and then you would also have the newly synthesized strand, and then that'll go into this. So this is essentially what a semi-conservative model is. Essentially, you're basically using the parental chromosome to make a copy of a newly synthesized chromosome, and that'll become the duplex right there for that. The conservative model, on the other hand, suggests that you would end up with a duplex that contains only the parental chromosomes, separate away from the newly synthesized chromosomes. So after replication, what will happen is that you would end up with two duplexes, one with the original parental strands and then one with newly synthesized chromosomes. Dispersive, on the other hand, is essentially you're getting a mix of newly synthesized and original parental strand in the same chromosome. So it could be something like this. So essentially that's what dispersive is. And that's essentially what most of the section is primarily going over. And that's basically it, to be honest. But the most difficult part about this whole entire section is understanding how this entire process will look like on a gel electrophoresis, and how you can determine a certain model of replication based on the bands that you're getting. So if you're not aware, a typical practice problem for these questions is essentially something like this, where you're given a prompt where where you have a certain organism, you're growing it in N15 media, and you transfer that organism to an N14 media, and then you extract the DNA and put it on the gel. And then essentially what you're trying to see here is how is this N14 how is that N14 being incorporated into the DNA? And that'll give you a clue on what kind of a mechanism of DNA replication is occurring in this organism. So yeah, what you would have is a gel. You would have a situation like this, where you have your ladder, N14, N15 mix, and then you have your N14. So you have like a gel like that.
And then typically what you would do is, this is referred to as, most of the time as the 0th generation or before. After you transfer it into the N14, you would take 2 samples out. You would take it after the 1st generation of replication and then the 2nd generation. So basically what you would end up here is you'll end up with 3 wells, 0, 1, and 2. And then because we've begun it doesn't matter what model we begin by, because we're starting first of all with N15, all of our samples should be at this particular well or this particular density, right? So after replication, and we can call this the semi-conservative model for the gel that I'm drawing right now, after replication, what we end up here is we end up with two different strands, or you end up with four homologous chromosomes. And essentially what happens here is that because both of these sister chromatids have the N15 and the N14, they would be considered as the hybrid. So each of these two will be considered a hybrid in terms of weight. So essentially what you would end up here is you would end up with a band that is located at the hybrid spot on your gel. So if we take that further, well, we have that, and we're trying to replicate that process through the semi-conservative model. What will happen here is that first of all you would conserve this strand over here, and then you would make a new strand. And then we're going to take that step forward with this one as well. We're going to conserve that blue strand, and we're going to add a new blue strand to it. Right? And then because we've originally had 2 of the original strands, you would just multiply this by 2. So basically 2. So you would have 2 of these. Because there's another one over here. So basically from this, what will happen is that after the 2nd generation, you have one that is hybrid and you have one that is one that is considered as N14. So basically what will end up happening here, you would have 2 bands, one at the hybrid spot and the one at the N14 spot, and that'll confirm that you have semi-conservative model of replication. So that is semi-conservative. The next one we're going to go over is we're going to go over the conservative model. So originally we always begin at the N15 mark right here. After the 1st generation of replication, we have 2 different bands or 2 different duplexes. One of these duplexes has the N15 and one of these has the N14. So you have 2 bands, one at the N15 mark and one at the N14 mark. And then what will happen here is after replication, again, you would end up with more N15 and N14. So you would end up with something like that. And then the dispersive, pretty simple. You're going to end up, because at the dispersive stage, you're basically making a hybrid all along, you're basically going to have 2 hybrids at both generation 1 and generation 2.
And then that's it.